with really practical stuff like AGI and, well, human regeneration.
It's definitely ambitious. We're looking at a hypothesis here that, frankly, challenges a lot of mainstream thinking about the body, the mind, and even how we should be trying to build true artificial intelligence or fix damaged tissues.
And the core idea coming from you, our listener, revolves around this concept of the quantum self. The suggestion is that the brain's map of the body, the neuromatrix, isn't just a passive picture, it's a quantum act.
Exactly. And this act is supposedly so powerful that it could sustain a real measurable field, a biophotonic field, in the space where a limb used to be.
So not just a feeling in the brain, but an actual detectable energy signature. Because the mind map insists the limb is still, there.
That's the core of the challenge to the standard view. The standard view is phantom limb sensations. That's purely neurological wiring gone a bit haywire.
Right, a short circuit.
Precisely. But this hypothesis asks, what if there's more? What if, by looking with the right kind of sensor, maybe some non-conventional light spectrum imaging, you could actually see biophotonic light coming from that empty space, as if the limb never left?
Okay, so it's like X-rays show bones, infrared shows heat. Is there a quantum self-spectrum? And the listener's thinking is, if we haven't found it, maybe we're just, well, not doing it right.
That's a powerful way to frame it. It pushes for innovation. But let's ground this a little first. Biophotons are real. Ultra-weak photon emission. It happens. Cells give off tiny amounts of light during metabolism. We can detect it.
Okay, so the basic phenomenon isn't controversial.
Not at all. It reflects cellular activity.
Yeah.
But when we look at the specific application here, testing this idea on amputees, we hit a wall.
How so? Has anyone actually tried pointing these sensitive detectors at an amputation site?
Well, here's the thing. There are, quite literally, absolutely zero studies measuring biophoton emission from amputees or at amputation sites.
Zero.
None. None reported in the literature. And standard imaging, like multispectral stuff, hasn't shown any weird electromagnetic fields hanging around in that empty space. The dominant view remains firmly neurological.
Okay, so that's the first major hurdle then, what you're calling the observation challenge. If this field exists, why haven't we seen it? Why is it so hard to find?
Because the signals, assuming they are there, would be incredibly weak, exceptionally faint.
Fainter than the normal biophotons we already measure.
Potentially, yes, or just completely drowned out. Think about it. The rest of the body is a constant buzz of biochemical activity, heat, other signals. It's like trying to hear a pin drop during a rock concert. This faint, hypothetical limb field is obscured by the existing biological complexity.
So it's a signal-to-noise problem, massively amplified.
Exactly. Finding a subtle, maybe complex spatiotemporal pattern against that overwhelming background noise, that requires a whole new level of detection and analysis.
Right. So if we accept the premise for a moment that the mind map is quantum, the output is there, how do we build the eyes to see it? What would this apparatus the listener envisions actually need?
We're talking about needing truly foundational technology, stuff that doesn't exist off the shelf. You'd need integrated systems. Things like arrays of tiny microscale chemical sensors.
Why chemical sensors?
For real-time metabolomics, to track the precise biochemistry happening moment by moment near the site, to filter out that noise. Then you'd need incredibly high-speed, non-invasive microscopy, something that can track tiny structures dynamically against the body's complex background.
And I guess the computing power to sort through all that data has to be immense.
Oh, absolutely. But it's not just raw power. The key is sophisticated signal processing.
Yeah.
Algorithms specifically designed to hunt for new kinds of complex patterns in space and time. Patterns that aren't just echoes of nerve signals or blood flow.
And that kind of toolkit, if developed, could potentially detect this hypothetical biophotonic limb outline.
That's the idea. And it links directly to the listener's other question. Could you then contain that output? Shape it?
Like build a scaffold that holds the biophotonic shape of the missing limb.
It's highly speculative, of course, but if you could reliably detect and characterize this field, the same tech might offer a way to interact with it, maybe stabilize it.
Oh.
Well, perhaps the apparatus could act like a waveguide or a resonator tuned to that specific biophotonic signature. It could, theoretically, provide a physical structure for the brain's quantum act to interface with. It bridges the mind map and potential physical regeneration, That's the kind of leap this substrate-focused thinking implies.
Okay, that's a huge leap. And it connects directly to this broader critique, right? That mainstream science, especially in areas like AI, is maybe too stuck on computation and ignoring the actual stuff life is made of. Trying to get complexity by just adding more processing power, doing it harder, instead of finding the right foundation, doing it right.
Exactly. And the whole debate around artificial general intelligence, AGI, is a perfect example. The dominant view, often called strong AI, is basically computationalism. The idea that mind is like software, it can run on any hard biological whatever.
Substrate independence.
Right. But then you have critiques like John Searle's Chinese room argument. It basically says a computer, even one that perfectly manipulates symbols, like translating Chinese, doesn't actually understand the meaning. It's all syntax, no semantics.
It's following rules, not comprehending.
Precisely. Searle argued for what he called biological naturalism, that consciousness needs the specific, unique causal powers of the physical brain. It's not just about the pattern of computation, it's about the material doing the computing.
So what makes biological matter so special then? What gives it these supposed causal powers that silicon lacks?
This is where we get to something really fundamental, the chirality mandate. Life on Earth is built with a specific handedness. Amino acids are all left-handed, L isomers. Sugars are all right-handed, D isomers. It's universal.
Like molecules having a specific twist. Why is that uniform twist so important? Just a fluke of evolution.
Doesn't seem like it. It appears to be a functional imperative. The sources argue that without this homochirality, build complex, stable biological molecules would be chaotic.
How so?
Imagine trying to build a zipper, but half the teeth are mirror images, they just won't mesh correctly. Homochirality ensures that proteins fold reliably, that DNA forms its double helix consistently. It prevents the chemical reactions needed to build long chains from just stopping randomly. It creates a stable, reliable, physical medium for emergent computation.
Okay, like having standardized parts is essential for building complex machines. Chirality standardizes life's building blocks.
That's a good analogy. It provides a necessary structural predictability. But it might go deeper than just structure. There's a specific quantum effect linked to this.
Ah, okay. Something beyond just reliable building blocks.
Yes, and this is potentially a game changer for the substrate debate. Chiral-induced spin selectivity, or CISS.
CISS. Okay, what does that mean in practice?
It means these chiral twisted molecules found in biology act like tiny filters for electron spin, even at room temperature.
Electron spin, like the electron's intrinsic magnetic orientation, up or down.
Exactly. So as electrons move through these chiral biological molecules, the molecule's handedness essentially forces the electron's spin into a specific orientation. It acts as a spin filter.
And silicon doesn't do that, not in the same way at room temperature.
Not intrinsically, no. This CISS effect is a concrete, measurable quantum mechanical property tied directly to the molecular structure of life. It provides a physical basis for arguing that biology does possess unique capabilities relevant to information processing, maybe even consciousness capabilities that pure computation on a different substrate might lack. It challenges that whole substrate-neutral idea head-on.
So the very handedness of life might enable quantum effects crucial for the brain's causal powers, maybe even for that quantum act producing the neuromatrix.
Fascinating. Okay, let's talk about the cascading effects. If we could solve that observation challenge, build those super-sensitive sensors and imaging tools, how does that suddenly help something seemingly unrelated, like regenerative medicine? Say, figuring out how lizards regrow tails.
The cascade is potentially huge because solving the observation problem gives us the missing tool. Think about regeneration. It's all about incredibly precise, subtle self-organization over time. Being able to see the metabolic activity, maybe even the biophotonic signals, in real time at the microscale. That's the window we need into that self-organizing blueprint.
We could finally track how the lizard pulls it off, and the sources mentioned specific genes involved in that lizard regeneration, right? Genes that are actually quite dangerous. That's right. Lizards manage to control some very powerful, highly conserved genes, essentially oncogenes, genes that drive growth. Things like WNT2B and EGFL6. These push cells to proliferate, build blood vessels, really accelerate repair.
But uncontrolled growth is cancer.
Exactly.
Yeah.
So the lizard balances these accelerators with crucial breaks, like the tumor suppressor gene ARHGAP28. They have the control blueprint to use these powerful tools safely, achieving scar-free regeneration. Humans have similar genes, but we lack that precise control manual. Unleashing them without it is incredibly risky.
So cracking the AGI code, If it does depend on understanding biological self-organization tied to chirality and quantum effects, that understanding of controlled self-organization is exactly what's needed to safely use those potent regenerative genes.
Precisely. Solving the AGI problem, if done through understanding biology's unique properties, could provide the blueprint for controlling complex biological processes like regeneration. The observation tools let us watch, the AGI insights potentially let us direct. They're two sides of the same coin, understanding and controlling biological organization.
It all comes back to understanding the substrate. Okay, one more cascade question about vestiges. The listener brought up the hiccup reflex, apparently a leftover from ancient fish trying to breathe with gills like 370 million years ago.
That's the leading theory, yes. A very old motor pattern.
So the question was, could we ever repurpose that reflex, maybe with some tech, to breathe underwater or filter feed? Is that plausible?
Plausible. Extremely unlikely. The sources put the probability at less than 5%, even over a million years.
Why so low?
It's a fundamental mismatch in physics and biology. Water is just so much denser than air, about 800 times denser. The pressure generated by a human hiccup, maybe 20 centimeter arrow, is nowhere near enough to efficiently move water through any kind of gill system.
Not enough.
Not even close. And for filter feeding, forget about it. A hiccup is intermittent. You might process, 150 liters of water an hour, max? A basking shark filters 1.5 million liters an hour. It needs continuous flow. The hiccup mechanism just doesn't scale biomechanically.
So, breathing water via hiccups is a non-starter. But then, why is this useless reflex even relevant to the bigger picture of AGI or life's origins?
Because its persistence is instructive. The fact that this ancient neural circuit for gill ventilation is still rattling around in our brainstem, even though it's now functionally useless for breathing water, is a perfect example of exactation, or perhaps just evolutionary baggage. It shows how deeply conserved these fundamental organizational circuits are.
The blueprint remains, even if the context makes it useless.
Exactly. And that supports the core idea relevant to AGI and abiogenesis. Ancient organizing principles are incredibly resilient. They stick around, they can sometimes be repurposed, or maybe just remain as echoes. It tells us that evolution builds on old foundations. Efficient blueprints don't just vanish.
Okay, so tying this all together, this whole deep dive seems to be pushing back against how science is typically done. What's the main takeaway for the listener who feels mainstream methods are too rigid, that they're missing what's possible?
I think the central message is this. Look, the difficulties we're facing with really big challenges, understanding consciousness, building true AGI, even radical regeneration. These might not be absolute prohibitions baked into physics. They might be limitations of our current tools, yes. What specific physical property does biology have that silicon doesn't? The sources point right at something fundamental to life itself.
Homochirality.
Handedness.
Exactly. Molecules that are mirror images but can't be superimposed, like your left and right hand, L&D forms. And life on Earth is obsessively universally homochiral. We only use L-amino acids for proteins and only D sugars for DNA and RNA.
And this isn't just a coincidence.
No, the sources argue it's a functional imperative. Life likely couldn't have gotten started or become complex without this absolute purity.
Why? What happens if you have a racemic soup of both L and D?
Well, the sources talk about computational chaos. It breaks down at the most basic levels. First, you get polymerization failure. Try building a long chain like RNA or DNA with a mix of left and right-handed sugars. The wrong-handed molecule acts like a poison. It stops the chain from growing. It poisons each other's polymerization. Think of trying to zip up a zipper where half the teeth are backward. Just jams.
Okay, so you can't build the long chains needed for information storage. What about proteins?
Same problem. Protein folding fails. Proteins need to fold into very specific 3D shapes to work as enzymes or structural components. That precise folding depends entirely on all the amino acids being the same handedness, the L form.
Like a very specific origami.
Exactly. Homochirality creates a narrow folding landscape, making those stable, functional shapes possible. Throw in a single D amino acid, and the whole structure can misfold, become useless, clumped together. So the emergence of homochirality is seen as this critical moment.
The point where chemistry becomes biology.
The moment a random chemical soup transitions into a viable physical medium for emergent computation, You need that specific physical property, homochirality, for complexity to arise.
And this physical necessity gets a huge boost from a really cool quantum effect,
Yeah.
CITSS.
Right, chiral-induced spin selectivity. This is fascinating. It turns out that chiral molecules, because of their twisted helical structure, act as natural spin filters for electrons passing through them.
Fin filters.
Yeah, an electron has spin, like a tiny magnet. When electrons move through these chiral molecules, their spins get aligned, polarized, and by a lot, like 60 to 90%, even at room temperature.
Okay, so how does that give biology an edge over silicon?
Well, think about how computers work now. It's all based on charge moving electrons around. Zeros and ones based on charge presence or absence.
Right.
But CISS implies that biological systems, being inherently chiral, can leverage electron spin. This potentially opens up access to low-energy quantum effects, ways of processing information using spin state that silicon just can't easily replicate.
Spintronics, but built into life itself.
Exactly. It could offer advantages in speed, energy efficiency, maybe even inherent parallelism or maintaining coherence. It strengthens Searle's argument for unique causal powers. Chirality might provide this essential thermodynamic tilt that helps biological systems self-organize and maybe even bootstrap, meaning semantics.
Which brings us back to the critique of how mainstream AI research often operates. If biology's secret sauce involves chirality and
Charge-based silicon models doing it harder might be fundamentally missing the point. It's ignoring the necessary physical medium.
so the alternative approach is
The synthetic abiogenesis idea. Don't just simulate intelligence, try to grow it. Bypass the Chinese room problem by creating the conditions, the right physical substrate, for understanding to emerge from the inside out.
like in this CL1 platform idea, a chemical soup on a chip.
Sort of. A self-assembling chemical system, likely chiral, interacting with a microelectrode array scaffold. The goal is to replicate the process that led to life and intelligence, not just copy the end result on the wrong hardware. It's about cultivating, not just computing.
Okay, let's shift gears now to evolution's own blueprints. From the origin of life to incredibly specific repair mechanisms. We need to talk about the Texas horned lizard.
Ah, yes, the blood-squirting defense. Amazing.
It is. It ruptures blood vessels near its eyes, squirts blood, and then the amazing part.
Yeah.
It repairs those vessels rapidly and perfectly, scar-free.
Which is the holy grail for human medicine.
Right.
Scar-free healing.
Totally. So if we want that ability, the lizard must have the genetic instructions. The sources point to specific genes, right?
Yeah.
What's the regulatory toolkit here?
Yeah, the core issue in humans is we tend to patch things up quickly with scars, prioritizing speed over perfection, probably to minimize infection. The lizard retains the ability to switch on controlled, perfect regeneration. The sources identify 4 key genes, and we have versions homologs of these in humans. We can think of them as accelerators and brakes.
Okay, give us the accelerators first. What drives the growth?
So on the go side, you've got genes that are actually known oncogenes, cancer-related genes in mammals. WNTTB is one. It's involved in cell proliferation, getting cells to divide to build the new structure.
An oncogene. That sounds risky.
It is. And the other accelerator is EGFL6, which promotes cell migration and the regrowth of blood vessels, angiogenesis, feeding the new tissue. Both are powerful growth promoters.
So activating these in an adult human.
Yeah.
Yeah, you'd need serious control.
Absolutely. Uncontrolled WNT signaling is linked to many cancers. That's why the control system, the brakes, is the real evolutionary genius here. There's ARHGAP28, which acts like a tumor suppressor. It helps control how cells move and divide, keeping things orderly.
Safety mechanism.
One of them, yes. And then there's BMPER, which acts as a fine-tuner for the whole process, especially controlling how those new blood vessels sprout and migrate. So the real secret isn't just having these genes.
We have the parts list.
Right. It's the lizard's incredibly sophisticated and safe regulatory network. It knows how to turn these powerful growth pathways on temporarily, locally, and then shut them down cleanly without causing runaway growth or cancer. That network is the blueprint for controlled self-organization, mastering growth without chaos.
That blueprint sounds crucial. Okay, let's pivot from that mastery of control to, well, something evolution seems to have left lying around awkwardly, the human hiccup.
Yes, the hiccup refresh.
Question came up, could this weird reflex, this evolutionary leftover, ever be repurposed? Could humans plausibly evolve to breathe underwater using it, maybe for filter feeding?
It's a great question because it highlights exaptation evolution reusing old parts for new tricks.
Hmm.
And the hiccup is an incredible neurological fossil.
How old?
The motor pattern dates back maybe 370 million years to our amphibian ancestors. It perfectly mirrors how tadpoles ventilate their gills.
Tadpoles. Seriously.
Seriously, a tadpole takes a gulp of water, then snaps its glottis shut, that little flap in the throat, forcing the water over its gills but not into its lungs. A human hiccup involves the same sharp intake of air followed by that same sudden 35 millisecond glottis closure. Same brainstem circuits, the CPGs, central pattern generators.
Wow. So we literally have a tadpole breathing circuit firing off randomly inside us.
Pretty much. It's solid proof of evolutionary conservation. The circuit's still there, even though its original function is long gone for us.
Okay, so the circuit exists.
Yeah.
Can we repurpose it for underwater feeding? The sources seem pretty doubtful.
Extremely doubtful. The probability is rated at less than 5%. The physical constraints are just too severe.
Like what?
First, water density. This is the killer. Water is about 800 times denser than air. You simply cannot move significant amounts of water using the gentle pressures involved in breathing air or hiccuping. A hiccup generates maybe 20 centimillijoules pressure. That's nothing compared to what's needed to pump dense water efficiently. Tadpoles use specialized buccal pumping for that reason.
So not enough power to move the water.
Not even close. Second, flow rate. Think about real filter feeders, like a basking shark or a whale. They process millions of liters of water per hour, continuously.
Right.
A human hiccup is just a brief spasm, maybe 4 to 60 times a minute. Even if you were hiccuping constantly and perfectly efficiently, you might process, 150, maybe 180 liters an hour? It's orders of magnitude less than what you'd need just to get enough calories from plankton.
Not enough volume either. Any other barriers?
Anatomy. We don't have the filtering structures, gill rakers, baleen plates. And our pharynx, our throat, is highly adapted for complex speech, not for separating water intake and outflow like a fish or tadpole.
Right. And the ultimate evolutionary hurdle, the trade-off.
The big one. To revert our anatomy back towards something potentially capable of aquatic feeding, we'd almost certainly have to sacrifice the descended larynx. That anatomical feature is crucial for the range of sounds needed for articulate speech.
Lose speech to maybe filter feed poorly?
It's an evolutionary non-starter. The cost is far too high. Evolution doesn't tend to rebuild complex lost structures, especially if it means giving up something as advantageous as language. The hiccup is a fascinating relic, but that's likely all it will ever be.
Or huge, seemingly separate challenges. The biophotonic self, getting AGI right with chirality, cracking lizard regeneration, and the dead end of underwater hiccups. Now, the key question, how are they connected? How does solving one potentially help solve the others? What's this cascading effect?
The deep connection lies in that shared scientific challenge we keep bumping into. The need to observe, understand, and ultimately controlled self-organization at very subtle biological levels.
Okay.
Whether it's the emergence of a quantum field, the development of consciousness from chemistry, or the precise regulation of tissue growth. A breakthrough in understanding or seeing that process in one area provides crucial tools or insights for the others.
All right, let's trace those cascades. Start with AGI. Let's say the synthetic biogenesis experiment works. The CL1 platform does show that consciousness requires this specific chiral biological substrate. What does that proof do for the phantom limb mystery?
It dramatically clarifies the situation. If consciousness is proven to be biologically bound, substrate-dependent, it effectively resolves that core philosophical debate that muddies the waters for AGI.
It kills substrate neutrality.
Right. And for phantom limb researchers, the message becomes crystal clear. Option A. The quantum self idea, the biophotonic field, is wrong because the neuromatrix operates purely classically, even if biologically unique. Option B. The biological substrate is key, and the only thing stopping us from seeing the quantum self is the technical barrier, the observation challenge.
So an AGI breakthrough forces the phantom limb question down to either as purely classical neurology, or we just need better instruments to see the biological quantum effect.
Precisely. It confirms biology is central and sharpens the focus purely onto the technological limitations of seeing it.
Okay, cascade number one. Now let's reverse it. What if we crack the lizard's secret first? We figure out exactly how it regulates those powerful oncogenes for perfect, safe repair. How does that help the AGI project?
It provides the missing blueprint for controlled self-organization. Remember, the core challenge in both the lizard repair and the CL1 abiogenesis experiment is managing these powerful, potentially runaway
Growth
processes.
signals.
Exactly. In the lizard, it's controlling WNT2B and friends. In the CL1 experiment, it's trying to kickstart a self-sustaining chemical reaction network, a kind of primitive metabolism without it just fizzling out or exploding into uncontrolled tar.
You need the same kind of biological guardrails for both. Initiation without chaos.
Yes. So figuring out the lizard's precise regulatory network, how it safely turns things on and off, gives the abiogenesis researchers a proven biological strategy. They can try to implement similar control principles, borrowed from evolution's playbook, to manage their synthetic system as it tries to bootstrap complexity and maybe, just maybe, intelligence.
Using nature's safety manual for building complexity. Makes sense. Now the big one. The technology itself. If we actually solve the observation challenge, develop those 4 key technologies, the microsensors, real-time chemistry, high-speed microscopy, smart algorithms, how does that cascade?
Oh, that's the game changer. Solving the observation challenge is the single most powerful enabling factor because it finally gives us the eyes to see what's really happening at these subtle biological levels.
It unlocks direct measurement.
Across the board. Suddenly, researchers could 1. Directly hunt for and measure those ultra-weak biophoton fields tied to the quantum self hypothesis. Test it properly, finally.
See if the phantom limb lights up, basically.
Essentially, yes. 2. They could watch protocells or other complex structures self-assembling in the CL1 experiment in real time, getting actual data on how chemical complexity might lead towards information processing or semantics.
Track the emergence of structure.
And 3, they could precisely track the release, diffusion, and decay of those growth factors like WNT2B in vascular repair models, confirming how the lizard's regulatory blueprint actually works moment by moment.
So the tech enables validation across all these frontiers.
It moves them all from the realm of hypothesis and indirect inference into the realm of direct observation and empirical testing. It's the foundational toolset needed for all this pioneer science.
Okay. And you even found a way to link the hiccup back in.
Well, yeah, just as a conceptual point, the hiccup, that repurposed tadpole circuit, is a beautiful example of exaptation. Evolution taking something old and finding a, in that case, accidental new context. It reinforces the idea behind the CL1 platform.
That it could be a directed abiogenesis engine.
Right. The hypothesis is that by providing the right chemical soup and the right environmental pressure, like the electrical stimulation from the MEA, we might be able to accelerate or repurpose some of life's conserved chemical organizing principles, like homochirality, maybe nudging the system towards complexity faster than random chance would allow. It's about harnessing evolution's toolkit.
So bringing it all together for you, the listener, it seems like we're looking at 2 really ambitious parallel paths emerging from these sources. On one hand, there's this philosophical push driven by ideas like Searle's and the necessity of chirality to pursue intelligence by actually growing it biologically. The bottom-up AGI garden, you could say.
Yeah, respecting the unique causal powers of biology. And on the other hand, there's this technical mandate, driven by the phantom limb hypothesis and the limits of current tools, to build entirely new kinds of sensors. Sensors capable of seeing the subtle physical effects the mind might have on the body, the biophotonic self.
And this really sharpens the critique of what the sources call rigid mainstream science. The argument isn't that current AI models or neuroscience aren't working hard enough.
It's not about just doing it harder with more computing power. The argument is maybe they're hitting walls because they're not doing it right. They're missing a fundamental biological factor like chirality, or they lack the observational tools to see the phenomena they're trying to explain.
Pioneer science needs that willingness to explore the unorthodox, right? To combine quantum ideas with synthetic biology, to take chirality seriously.
Exactly, because we know life did emerge from a chaotic chemical soup, and that process was fundamentally chiral. And the only thing we know of that produces complex consciousness, the brain, is also chiral.
Which leads us to that final, really provocative question to leave you with.
If we could somehow create a perfect mirror image version of life, build organisms from D amino acids and L sugars instead of our L amino and D sugars, would that mirror life be conscious just like us?
Or would it be a perfect biological machine, a functional zombie, lacking that specific chiral structure, that thermodynamic tilt may be needed to bootstrap real understanding, real semantics?
And maybe even more critically, even if we could build that mirror life, if we haven't first solved the observation challenge, if we haven't built those revolutionary sensors,
Would we even be able to tell if it was conscious or not? Or would its inner reality, whatever it might be, remain completely invisible to us, lost in the biological noise?